Contribution of genetic variants and ischemic stimulus to human myocardial transcriptomics. Inter-individual gene expression variation is impacted by both genetic variation and environmental exposures. We investigated genetic transcriptomic relationships at baseline and after exposure to myocardial ischemia as an environmental factor to understand mechanisms that underlie the pathological processes of myocardial ischemia in humans. In Figure 1, we see a study design and analysis workflow. In this study, we have two different cohorts. The left side left is a study of left ventricular biopsies from 118 patients undergoing aortic valve replacement surgery. And we obtained the biopsies at baseline and after an average of 79 minutes of ischemia. For the analysis, we focused on ccQTL. We explored the genetic basis of gene expression through mapping eQTL at baseline and post-ischemia after exposure to ischemia, focusing on ccQTLs where gene expression levels are affected by variants in the gene body and one megabase flanking the gene. We identified ccQTL at FDR smaller than 0.05, which was equivalent with 1 E-6 in our study. Some of the results are reviewed in Figure 1. In the left side, we have a Venn diagram of genes with at least one significant ccQTL. The overlap, which are 1,265 genes, represent the number of genes with ccQTL at both baseline and post-ischemia conditions. We have more than 600 genes that are eQTL at baseline but are not eQTL after exposure to ischemia. In contrast, more than 400 genes that were not eQTL at baseline were eQTL after exposure to ischemia. In the right side, I am comparing the ccQTL effect sizes, baseline versus post-ischemia conditions. The large effect sizes, red dots, belong to ccQTLs common at both conditions. The similar ccQTL effect sizes, the smaller ccQTL effect sizes belong to either baseline specific condition, blue dots, or post-ischemia specific conditions, green dots. The effect sizes are distributed around line 45 degrees, which means the effect sizes do not change dramatically after exposing to ischemia. And no ccQTL change direction between baseline and post-ischemia conditions because there is no dot in either upper left or lower right of the plot. In another attempt, we focused on mechanistic understanding of post-ischemia eQTL in myocardial ischemia injury. We had another study that we had troponin levels, which is a biomarker of cardiac injury, recorded, and that was the study of 1,751 patients undergoing a cardiac surgery. And in this study, we had genotyping data, but not gene expression levels. Therefore, we used the summary statistics from the eQTL study and then predicted the gene expression in the GWAS study. And then we investigated the relationship between genetically predicted genes and troponin levels. This is called transcriptomic wide association study. We also assess the assumptions of Mendelian randomization to identify the causal genes of troponin level. The result was 19 cis-regulatory genes that acting as mediators of myocardial infarction effects.